Usually, when we talk about a medical diagnosis, there's this expectation of absolute precision, like engineering.
Right, like it's a math problem.
Yeah, exactly. You break your arm, the X-ray shows that jagged white line, and the doctor just points and says, there it is, broken.
It's very binary. The bone is either intact or it's fractured.
Right, and it's the exact same logic with our electronics. Like, I'd like to imagine the optic nerve as this incredibly thick fiber optic cable.
That's a great way to picture it.
Yeah, and it connects a high-tech camera, which is your eye, obviously, to a supercomputer, your brain.
Sure.
And if that cable gets completely sliced in half, the screen goes dead, period.
Yeah.
Up until now, you couldn't just splice biological wires back together. The hardware was permanently broken.
It really is the absolute definition of a biological dead end. I mean, historically, the entire goal in neurobiology was something we call anatomical restitution.
Anatomical restitution. So trying to put it back exactly how it was.
Exactly. The dream was always to find some way to coax the original host axons, those original wires from the eye, to just magically regrow across the empty void.
It'd just reconnect perfectly to the brain.
Right. But in the central nervous system, and specifically the optic nerve, that simply doesn't happen. The body actually actively prevents it.
Which is exactly why the source material for today's deep dive is so mind-bending. Our mission today is to thoroughly unpack a truly groundbreaking 2026 paper from Cell Stem Cell.
Oh, the Gong et al. Paper. It's incredible.
Yes, by Gong and colleagues. We are looking at a completely severed optic nerve, a condition universally considered irreversible, being repaired using human lab-grown retinal organoids.
Which is just, it's sci-fi level stuff.
Right. Think of it like inserting a biological universal adapter between two cut wires.
And what's fascinating here is the massive paradigm shift, right? These researchers aren't just stubbornly trying to force the original nerve to regrow the way we always have.
They gave up on the anatomical restitution.
Exactly. They are building a chimeric relay system. They're accepting that the original continuous wire is broken, and instead they're intentionally installing a biological middleman.
Okay, let's unpack this, because before we can understand the fix, we really need to understand the break.
Yeah, the injury model is crucial here.
Right, and I want to get into the exact anatomy of this adapter. What exactly are we looking at in this rat model? Because they didn't just nick the nerve, right?
No, not at all. We're looking at a complete devastating injury. The researchers made a full transection of the optic nerve exactly 1.5 millimeters behind the eyeball of the rat.
Okay, 1.5 millimeters.
Yeah, and they didn't just cleanly slice it. They actually removed a 0.45 millimeter segment of the internal nerve tissue.
Wait, they removed a chunk?
They did. They left a standardized, completely empty gap of pure white matter inside the nerve. They stripped the parenchyma out entirely, so the connection is entirely gone.
A completely empty void. And into that microscopic gap, they placed the adapter. Let's talk about that, because we aren't talking about some synthetic polymer or plastic tubing here. This is living human tissue.
Exactly. They used human retinal organoids, or hROs, which are derived from H9 human embryonic stem cells.
Stem cells, okay.
Yeah, and by day 30 of their development in the lab, these microscopic three-dimensional structures are essentially tiny developing human retinas.
Wait, let me make sure I'm getting this. They are dropping a tiny 30-day-old human eye tissue into a severed rat nerve.
Basically, yes.
Is it just a random clump of cells?
Not at all, actually. It's a highly structured ecosystem. They made sure this organoid contained different, highly organized layers of developing cells.
Like what kind of layers?
Well, on the outer layers, you have specific progenitor cells. They're marked by proteins called CHX10 and PIAX6. Think of these as blank slates that are structurally ready to become retinal tissue.
Or CHX10 and PIAX6 on the outside.
Right. But deeper inside, you have ATOH7 positive cells, which are slated to become retinal ganglion cells, the actual cables of the optic nerve.
Oh, wow.
Yeah, and some of them are already fully mature, differentiated cells expressing BRM3B and PUJ1 markers.
So it's essentially a customized microscopic bundle of human wiring, about, 0.5 cubic millimeters in size.
Exactly 0.5 cubic millimeters.
And they take this tiny human organoid and perform a procedure they call hROITON, which is a mouthful.
It is a mouthful, yeah.
It stands for Human Retinal Organoid Intrathecal Transplantation into the Optic Nerve. And they use a drug called cyclosporine A, right?
Yes, cyclosporine A is an immunosuppressant. It prevents the rat's immune system from rejecting the human tissue.
Right. But hang on, I have to push back here. You're taking a microscopic ball of soft human tissue, and dropping it into the severed, bleeding nerve cavity of a rat.
I know, it sounds impossible.
How does it not just get crushed or turned to mush?
That is the absolute brilliance of the surgical design. They didn't just drop it into an open wound. The optic nerve has a natural outer casing, a sheath. When they created that 0.45 millimeter gap, they only cleared out the internal wire, the parenchyma, but they kept that outer sheath completely intact.
Oh, so the casing was still there.
Exactly. That sheath acts as a perfect natural physical sleeve. It holds the soft, human organoid securely in place, perfectly aligning it with the cut ends of the rat's nerve without crushing it.
Ah, that makes perfect sense. It's literally like slipping a new piece of exposed wire into a piece of existing heat-shrink tubing to hold it steady.
That's a really good analogy.
So to pull this first part together for you listening, let's do a quick recap. We have a 0.45 millimeter physical void in our rat's nerve, and the researchers used the hROITON procedure to slide a highly organized 30-day-old human tissue adapter into the intact sheath to bridge the gap.
Spot on.
So it really makes you wonder, as you're taking this in, if you were tasked with designing a biological bridge for the brain, would you prioritize the rigid structural strength to hold the gap open, or the cellular flexibility to allow for new electrical connections?
And that tension between physical structure and electrical function is exactly the hurdle the researchers had to measure next.
Right.
Because once that adapter is in place inside the sleeve, the immediate critical question is, does the surrounding hardware actually survive the trauma of the surgery?
Yeah, because a physical cable can look fine on the outside, but if the camera it's plugged into is broken, it doesn't matter. So did it look right structurally?
To figure that out, they used optical coherence tomography, or OCT.
OCT, right. I've heard of that.
Yeah, it's a clinical imaging tool, very common in ophthalmology. It uses light waves to take incredibly detailed microscopic cross-section pictures of the retina.
And what do the scans show at day 30 after the surgery? I assume this is post-surgical day 30.
Yes, PSD 30. The contrast was striking. In the control group, the rats with the severed nerve but no human adapter, the hardware was essentially withering away from lack of connection.
Like it was starving.
Pretty much. The optic disc looked incredibly pale. The inner layers of the retina were entirely disorganized, and the ganglion cell complex, or GCC, was severely thinned out.
That's the layer responsible for sending the signal down the nerve.
Exactly. And if you looked at the gross anatomy, the physical tissue of the severed nerve had literally liquefied.
Liquefied. Wow. That is a horrifying but very clear picture of a dead biological wire.
It really is.
But what about the group that got the human organoid adapter?
A completely different architectural story. The grafted group maintained a healthy, pale red optic disc. The thickness of their GCC retinal layers was preserved.
Oh, wow.
And physically, when looking at the nerve itself, it didn't liquefy, it was contiguous. It maintained this healthy porcelain white color spanning the entire gap.
Porcelain white. So the human organoid physically rescued the structural integrity of the rat's eye.
It did. Structurally, it was a massive success.
Okay, that's an incredible physical rescue. And here's where I get skeptical.
Okay, let's hear it.
A plugged-in cable can be contiguous. It can look completely whole, porcelain white, perfectly healthy from the outside, but carry absolutely no data.
That's true.
It can just be a dead connection.
Yeah.
So structurally, it survived, but what does this actually mean for sight?
You're completely right to ask that. Preserving the physical architecture is a strict prerequisite for function because, if the tissue liquefies, the electricity has nowhere to travel.
Right.
But architecture alone doesn't guarantee the transmission of data.
Yeah, but bridge is useless if no cars can drive across it.
Exactly.
So, recapping the architecture for you. Without the graft, the severed nerve liquefied and degraded. With the graft, the nerve remained whole, preserved the GCC, and looked physically healthy on the OCT scans.
Perfectly summarized.
Which brings up the next massive hurdle. Why is preserving the physical architecture of the nerve just as important as fixing the internal wiring? Well, without the road, there's no traffic. But how do you actually prove that visual data is traveling across that bridge and reaching a rat's brain? You can't exactly ask a rat to read an eye chart.
No, you definitely can't. You have to rely entirely on their physiological reflexes. And the researchers designed an absolute gauntlet of rigorous tests to prove it at PSD 30.
What kind of tests?
Well, first, to ensure no cheating, they physically sutured the rat's uninjured, healthy eye shut.
Suture it shut.
Yeah, and covered it with a black mask.
Wow. Okay, so every single visual response they measure absolutely has to come exclusively from the eye with the severed spliced optic nerve.
Exactly. The first test was the optomotor response, or OKR. They placed the rat in a specialized chamber where the walls are covered in vertical stripes that slowly rotate.
Oh, I've seen videos of this.
Yeah, it tests the accessory optic system. If a rat can visually perceive the moving stripes, it will reflexively move its head to track them.
And did they track them?
They did. Uninjured rats have a visual acuity score of about 0.529 cycles per degree in this test. The rats with the completely severed, untreated nerve scored exactly 0.
Total blindness.
Right. But the rats with the human adapter recovered a visual acuity of 0.137.
Well, 0.137.
Yeah, and their contrast sensitivity recovered to 1.377. It is absolutely not perfect high-definition vision, but they were unquestionably tracking the physical movement.
That's wild. But the test that I found even more fascinating in the paper was the pupillary light reflex, the PLR.
Oh, the PLR test was brilliantly designed.
They shined a very specific 622 nanometer red light into the eye at 25 lux. Now, I had to ask, why not just flick on a flashlight or the room lights to see if the pupil shrinks?
Yeah.
Why such a hyper-specific shade of dim red light?
Because the researchers are being incredibly meticulous. If you just hit the eye with standard bright white light, you trigger every single pathway at once.
Right.
That includes a specialized set of retinal cells called iPRGCs that react to blue light, peaking around 480 nanometers, which manage your circadian rhythms.
Okay, so the blue light is a different pathway entirely.
Exactly. By using a dim 25 lux, 622 nanometer red light, they specifically bypassed those other systems. They avoided over-triggering the rods and cones, isolating the exact visual circuit they wanted to measure.
That is just brilliant experimental design. And the results?
Over 52%, specifically 52.17% of the grafted rats showed a direct pupillary reflex.
Wow.
Yeah, the pupil physically constricted when that specific red light hit the retina. The reaction time, the latency, was slightly delayed compared to a healthy eye, but the final tightness, the amplitude of the constriction, was totally normal.
Half the rats regained their biological light reflex through a completely severed nerve.
It's incredible.
But they didn't just stop at reflexes. They went a layer deeper into the actual electrical physics of the nerve, right? Electrophysiology.
Yes, they measured something called FONP, flash-induced optic nerve potentials. They attached sensors to literally measure the raw electricity moving down the biological cable when light hit the eye.
And what are they looking for there?
They're looking for specific electrical peaks called P1, N1, and P2. In an uninjured eye, you see these very clearly.
And this is where I think about my old dial-up internet from the 90s.
Oh, this is a great analogy.
Because in a perfectly healthy, uninjured rat, that electrical signal comes in at about 14 microvolts. That is your high-speed fiber optic broadband.
Exactly.
In the untreated severed nerve, it's 0, dead air. But in the grafted rats, the electrical signal, those P1, N1, and P2 peaks, returned. But the voltage was only 1.94 microvolts.
Right, 1.94 compared to 14, it's a fraction of the original bandwidth.
Exactly. It's a screeching 56K dial-up modem.
Yeah.
You are definitely not streaming 4K video on 1.94 microvolts.
Definitely not.
But it is enough bandwidth to send an email. It is enough voltage to send the fundamental critical message of light versus dark to the visual center of the brain.
That's a perfect way to put it. The signal is degraded, yes, but the electrical circuit is undeniably closed.
So summarizing this functional data for you, they used the rotating stripes in the OKR to prove spatial tracking returned to 0.137. They used the isolated red light to prove the PLR reflex returned in over 52% of subjects. And they measured FONP to prove a 1.94 microvolt signal was physically traveling down the cable.
Really solid summary.
But thinking about that 52% success rate in the reflex test, it makes you wonder, based on a 52% success rate, what variables do you think make the difference between a successful graft and a failed one in the other 48%?
And asking those kinds of skeptical questions is exactly what drives the scientific method, because the researchers had to play their own devil's advocate here. Think about it. How do we know for absolute certain that this visual recovery is actually because of the human cells?
Wait, what else would it be?
Well, what if dropping that tissue in there just caused the rat's own nervous system to panic, release a bunch of generic growth factors, and the rat experienced some miraculous self-healing?
Oh, I see. If I'm a skeptic, I'm saying the human tissue was just a glorified Band-Aid that prompted the rat to fix itself.
Precisely.
So we need a way to prove the human adapter is doing the heavy lifting. We need a way to literally turn the human cells off and see what happens.
Which is exactly what they did. They installed a biological kill switch.
A kill switch?
Yes. They used an incredibly advanced chemogenetic technology called DREADDs.
DREADDs. I love when scientists force acronyms to spell cool words. What does it stand for?
Designer receptors exclusively activated by designer drugs.
Okay, that sounds like science fiction. How does a biological kill switch actually work?
It works like a lock and a key. Before they ever transplanted the human organoids into the rats, they used a harmless virus, an AAV vector, to genetically modify the human cells.
An AAV vector, got it.
Right. They inserted a specific gene called HM4DI. This gene forced the human cells to grow a specialized receptor on their surface.
So that receptor is the lock.
Exactly. But here's the genius part. That artificial lock does absolutely nothing in normal daily life. It sits there, totally inert. The human cells function normally. They transmit the visual signals the rat can see. The lock has no key in the natural world.
Until the researchers introduced the custom key.
Yes. The key is a highly specific synthetic designer drug called DCZ-dichloroclozapine.
DZ.
When they inject the rat with DCZ, the drug circulates, finds those specific artificial locks on the human cells, and binds to them. And when it binds, it acts like a chemical handbrake. It forcibly stops those human neurons from firing any electrical signals. It mutes them.
That is wild. You essentially install a remote control mute button that only affects the human cells inside a rat's head.
Exactly.
And the results of hitting that mute button.
They were instant and undeniable. The moment DCZ was injected, the human cells went completely silent. We know this because the activation of CFOS, a marker of neural activity, dropped from 56% down to just 13%.
Wow, so it plummeted.
It tanked. And the second the human cells went offline, the rat's cortical signals measured by FEEP and the firing rates in the brain's superior colliculus just flatlined.
And the behavioral test reflected that flatline too, right?
Completely. They ran a visual cliff test, which evaluates depth perception, and a looming test, where a dark shadow expands quickly, mimicking a predator swooping down.
Like a hawk or something.
Right. Before the drug, the grafted rats reacted perfectly. They recognized the drop-off, they hid from the shadow. But the moment the DCZ drug hit the human cells, those survival behaviors completely vanished. They became completely blind again.
So if muting the human cells instantly blinds the rat, the human cells absolutely must be the physical wire.
There is no other explanation.
There is no miraculous rat self-healing. The human tissue is the bridge.
It is the ultimate airtight proof of concept. The entire visual recovery is dependent on the human graft actively transmitting the signal.
So recapping this for you, we have a genetically engineered lock via an AAV vector on the human cells, and a chemical key DCZ that acts as a handbrake. Injecting the key silences the human adapter, dropping CFOS from 56 to 13%, which instantly erases all vision and behavioral responses.
Nailed it.
It proves the adapter works. But it also makes you imagine, what are the broader applications of this dread technology in human medicine? Imagine being able to swallow a pill that acts as a temporary pause button for a very specific, hyperactive neural circuit causing chronic pain or seizures without affecting the rest of the brain. Where else could a biological pause button be useful?
The level of targeted control it offers bioengineering is staggering. But even with the kill switch proving the human cells are doing the work, we still have one final, massive, missing piece of the puzzle.
Okay, what's missing?
We know the architecture is saved. We know the data transmits. We know the human graft is responsible. But how are the individual wires actually connecting?
Right. Like, did the rat's original eye just grow one single, super long, continuous wire all the way through the human tissue, using it like an empty conduit, and plug directly into its own brain?
To figure out the exact physical mechanism, they used an anatomical tracing technique. They injected a fluorescent tracer called cholera toxin B directly into the rat's eye.
Cholera toxin B.
Yeah, CTB. This tracer physically travels down the internal wiring, basically drawing a glowing microscopic map of the neural pathways.
And what did the glowing map reveal? Did it go all the way through?
It revealed the non-canonical relay. The rat's original wires did not grow straight through the human adapter. Instead, the glowing map showed a comb-like distribution of the rat's axons reaching into the human graft and stopping.
Stopping? Then how does the signal get to the brain?
Well, they rebundle at the optic chiasm and reach the superior colliculus because they formed synapses.
Oh, they form a connection.
Yes. A synapse is a junction where one neuron chemically talks to another. The researchers found presynaptic markers, specifically synapsin-1, which is the sending side of the connection, from the rat physically touching postsynaptic markers called PSD95.
And where were those postsynaptic markers?
They were located entirely on the human graft cells, which they identified using a marker called STEM121 plus BABO.
STEM121 plus SBIO.
The rat tissue and the human tissue were forming literal, functional, microscopic connections.
I love this. Think of it exactly like a relay race on a track.
Oh, that's a perfect visual.
The rat's eye is runner number 1. It carries the baton, which is the visual electrical signal, and runs right up to the edge of the severed gap. It hands the baton off to the human organoid, which acts as runner number 2.
Right.
The human cells carry that electrical signal across the void and then hand it off to the rat's brain, which is runner number 3. The human tissue isn't just a passive tube. It's an active biological participant actively handing off the data.
It's an incredibly dynamic process. And what's equally astonishing is how the rat's own immune system supported this relay race.
What do you mean?
Well, normally, when the central nervous system suffers a massive injury like this, the body's immediate defense mechanism is to form a glial scar.
Let me guess. A scar in the nervous system is a bad thing.
It's a total disaster for regeneration. Specialized cells called astrocytes marked by GFAP plus rush to the site of the injury and build a dense, impenetrable wall of scar tissue.
Like a barricade.
Exactly. It's the body's way of walling off the damage to protect the rest of the brain, but it physically blocks any new wires from ever growing through.
But the tracer map showed something different here.
Completely different. When they looked at the genetic activity of the rat's cellular builders, those GFAP plus astrocytes, they saw something incredible. Instead of panicking and forming a thick barrier, the host's astrocytes adapted.
Adapted how?
They actually formed highly ordered, microscopic, bridge-like structures spanning the gap. They were literally building scaffolding to actively guide the rat's axons toward the human tissue.
They built a bridge instead of a wall.
Exactly. And furthermore, using advanced single-nucleus RNA sequencing, or snRNA-seq, on the local immune cells, they proved that the rat's natural defenders, the microglia and T cells, basically calmed down.
So no rejection.
Right. There was no runaway inflammation or overt immune rejection. The rat's body fundamentally recognized the human adapter as an ally, not an invader.
That's just wild. So, looking at the entire mechanism for you. The CTB fluorescent tracer proved this isn't one long wire. It's a 3-part synaptic relay race from rat to human to rat, confirmed by those synapsin-1 and PSD95 markers.
Yes.
And the host's immune system completely adapted, building a GFA plus microscopic guiding bridge instead of a defensive scar. When you hear that, it really challenges your assumptions. Does knowing that highly complex visual data can function perfectly well through a biological middleman completely change how you think about the hardwiring of the brain? It's far more plastic and adaptable than we ever thought.
And that profound adaptability leads us to the grand so what of this entire paper. This represents a monumental shift in how we approach neurobiology.
Right.
We are officially moving away from the rigid, impossible demand for strict anatomical regeneration, where we just pray the original wires reconnect perfectly. Instead, we are entering an era where we can engineer functional chimeric waste stations.
Waste stations, I like that.
Yeah, we can build composite, multi-species biological structures that successfully restore signal transmission.
It's an incredible breakthrough. But, you know, as with all cutting-edge science, we have to ground this in reality. We can't just say we've cured blindness tomorrow.
Yeah.
What are the actual limitations of this specific study?
We have to be very clear about the limitations. First, as we discussed with the dial-up modem analogy, this is a non-canonical relay. It's a 56K connection.
Right.
We have absolutely no idea if the rat is perceiving a clear, high-definition picture of the world, like the sharp outline of a piece of keys, or if it is merely sensing raw gradients of light and motion.
Right. Recognizing a shadow moving is very different from recognizing a face.
Exactly. Furthermore, the anatomical scale is vastly different. A human optic nerve is infinitely longer, thicker, and vastly more complex than a rat's.
Obviously.
Engineering a human-scale organoid adapter to perfectly hand off millions of signals across much larger gap while navigating our incredibly complex human immune system over a lifetime that is a massive, towering hurdle.
It really is the difference between splicing a wire for a desk lamp and trying to rewire the electrical grid of a skyscraper. The scale changes the math entirely.
It really does.
But the proof of concept is undeniably there. The biological screen turned back on. And I want to leave you, our listener, with a final provocative thought that builds on exactly the mechanism we've learned today.
Oh, this is a good one.
We spent this entire time talking about the optic nerve, which is pure white matter in the central nervous system. But if human, lab-grown organoids can be successfully engineered to act as a synaptic adapter bridging a severed optic nerve, could we eventually customize similar organoids to bridge severed spinal cords?
Wow.
Could we build localized biological routers to relay signals past catastrophic spinal injuries, effectively restoring movement and sensation to paralyzed limbs?
I mean, if the central nervous system is fundamentally willing to accept a middleman relay, the possibilities for restoring human function are truly profound.
It is definitely something to mull over. Thank you for joining us on this deep dive. Keep questioning the boundaries of human biology, and we'll catch you next time.